And I want to welcome everybody again for our extended year-long annual meeting this year. As you know, in the January time slot, we didn't get enough people to make it worthwhile for all of our speakers. And so we've extended this to be the whole year long. And as a result, you get more time with each speaker, and we get more time for questions and so on. So we're looking forward to this format. Today, there's two talks, and there will be this one by Hubert, and then Wayne will come on in about an hour to give his talk. And we will sign out of this link because it is not the same link for the two talks. So we'll be ending this meeting a couple minutes before the top of the hour, and then you'll all be signing in if you wish to come to the new talk. Mary or Kat will put in the chat the link if you want to sign up for the second talk if you hadn't done so already. I'm also going to remind you that what we've asked our researchers and our clinicians to do is to talk about PXE from their point of view, which means that in some sense it's a piece of the pie, a piece of the puzzle. And just like any single piece of the puzzle, it doesn't tell the whole story. And particularly with the research talks, we remind you that nothing that is being said here is a approved treatment for PXE. And so after these talks, I often get many questions. Where can I buy that? Can I go take it? How much of it should I take? Should I be doing that? Whatever the talk was about. And the answer to all of this is no. We should not be playing with taking anything until it's approved after rigorous clinical trials, despite even some of the enthusiasm of some of our researchers. So I know that Hubert will be much more moderate in his comments around not over-promising and not hyping, although we have a great deal of hope and we're really excited by the advances over the last now 29 years that PXE International has been around. So we're pretty happy with that. So without further ado, I'm going to introduce to you Dr. Hubert Chou, who is the Executive Director of Clinical Development at Daiichi Sankyo, which is a large pharmaceutical company. He's located in New Jersey. The company is Japanese, and we're very delighted to have worked with him for a very long time now, and very excited by his discoveries and development of therapies, and also his commitment overall to PXE really has been remarkable and very wonderful to have him on our team. So without further ado, Hubert, if you have questions, put them in the chat. We will have time at the end of the talk for questions, and you can either put them in the chat then, or you can unmute at that point. Thank you.
All right. Well, thank you very much, Sharon, for the nice introduction. Let me share my screen. All right. Again, thank you for the, let me go ahead and get started on this discussion. First of all, a full disclosure, I'm an employee of Daiichi Sankyo, and what I'd like to discuss with you is share my views about the clinical trial and the clinical development process for a new medicine for PXE. And as part of this effort, that jointly with all of you, this one note is that Daiichi Sankyo is developing a new medicine for atopic calcification that includes PXE. So let's go back to what PXE is. This is it's a slide that you have seen many times before. Certainly PXE is a multi-organ systemic calcification disease, and really just use this slide to say that there are so far no approved therapies specific for PXE. And we know there are a lot of the effort has gone into understand this pathogenesis of this disease, including the discovery of ABCC6 and the role of pyrophosphate. And the next slide is really just a way to capture these molecular level of disease mechanistic pathway, say that the ABCC6 is, as we understand it today, is where the defect is, that they reduce the ATP, which subsequently process to generate pyrophosphate, which is a potent inhibitor of ectopic calcification. And that's through the enzyme EMPP1 and the subsequent steps in further processing of both the pyrophosphate AMP and the role that the TNAP in pyrophosphate degradation. And through that understanding, what will lead to further is these agents potentially these molecular target becomes a drug target. Besides the TNAP inhibition that we can use, there are other efforts in targeting the ABCC6, in targeting the EMPP1, or in including using exogenous pyrophosphate, all leading to hopefully reducing the ectopic calcification itself. And so, and many of you know, there have been a lot of the effort besides the basic science research moving to the clinical phase. And Sharon had mentioned a few times in the last few years now that we're really in the exciting phase of the clinical research for PXE. There have, in the clinicaltrial.gov page, you can find these clinical trial information, including the one completed several years ago, the magnesium trials. There are also the anti-VEGF for treating the retinal complication of PXE. There are the efforts in using etiginate to prevent the ectopic calcification. Pyrophosphate, as I mentioned, it's been studied how that may reduce the calcification also. Our colleagues over at Inozyme are using the EMPP1, modified EMPP1 enzyme for people with the ABCC6 defect. And certainly there are also the other ways to target and to increasing organic phosphate, including the TNAP, the agent that has TNAP inhibitor activity, including the effort by Daiichi Sankyo with the DS-1211. But long before these studies can be done, and particularly for a new chemical entity, there are a lot of things in the preclinical development stage that need to be done, including the understanding of biology, the pharmacology, understanding what these new chemical entity that you want to study, what kind of property it has, something called ADME, or absorption, distribution, metabolism, and elimination. Their safety obviously is very important for all of us, and that's why there's in the preclinical space a very large effort in understanding the toxicological profile of these compounds. And lastly, with the new chemical entities, we simply need to formulate into a suitable medication dosage form. Right. So let me take these one step at a time. In the preclinical biology, pharmacology step, as you have heard over the years, many of the efforts from either the understanding the role of pyrophosphate to whether the pyrophosphate can be used to the retinal disease pathophysiology, to using the knockout mice, for example, or both the in vitro, in vivo, and also the continued effort you recently heard from Olivier Lassauce and from Lily about the genetics as well. So these effort in preclinical research in pharmacology and biology really go to providing evidence supporting any of these targets, whether it's pyrophosphate, etiginate, it can be really used, and pyrophosphate is the target of choice in developing a new medicine. So beyond understanding that, and let's assume we know what the target is and how we're going to be approaching it, and we develop and identify a new chemical entity that potentially can be developing a drug. We need to really understand what that chemical is and how is that getting into your body. This is through the assessment of ADME, or the absorption, distribution, metabolism, or elimination. What they really say is how this drug, does it get into your body if you administered either by oral route or by whatever route that you of choice. And once it get in, where does it go? And what does your body do with it? Does it transform it? Does it digest it, metabolize it? And lastly, you don't want the chemical entity to be staying in your body for long. And so your body need to remove, and how does your body find ways to get rid of it, either through your kidney, through your liver, or bile, or other ways. And particularly this metabolism part, it's a very complicated and very involved investigation by the preclinical scientists. And not to go into the specific details, but you can see the illustration here, how the different metabolic enzymes in your body go through these chemical transformation of the compound. This is something very important and understanding to have. This is once you have all that. As I said, safety is very important, and the preclinical toxicology evaluation, particularly this illustration here is for small molecule compounds. There is a large amount of investigation that you need to do, or we need to do, to understand these chemical entities. And in the preclinical space, before even putting the drug into human body, once a particular candidate is selected, there's a battery of tests, including genotoxicity study, safety pharmacology study, and for the different specific organ system. There's short-term toxicology study in 2 different species, usually rodent and non-rodent, for 2 to 4 weeks. And there are other battery of tests need to be done. Once you finish all that's all completed, and you identify a safe dose, you can file the IND, or the investigational new drug application, and to allow you to go into the clinical testing. The toxicology evaluation does not stop once you go into the human. And there will be increasing longer duration of study that need to be done, including the subchronic toxicity study up to 3 months, and chronic toxicity study after essentially a lifetime of these animals, rodent in 6 months and non-rodent 9 months. There are others, including reproductive toxicology, carcinogenicity, juvenile toxicity in the developing animal. And again, usually it's done with 2 different species, at least 1 rodent and one non-rodent species. With all that, let's assume we finish. You cannot really ask us to be taking a spoonful of powder, or what's called active drug or active pharmaceutical ingredient. These substance, a new chemical entity, need to be formulated into a suitable drug form, such as a tablet, capsule, or an injectable, if that's the case. And that's usually done through mixing with a suitable excipients that combine into the tablet. And through those efforts, and this is what we see in the mature or marketed product form. These processes are very detailed and regulated, and need to provide the consistency so that there is a large effort, something called CMC, chemistry, manufacturing, and control, that really detailed out and have every single batch follow exactly how it's put together, what ingredient it is, how it's put together, and how the manufacturing process is done. All right. So let's assume you have all that. Then what? How do you go into clinical trials? And what is clinical trials? Clinical trials is really a process to evaluate drugs, devices, or medical procedure and really to assess is the safety and effectiveness. And trials are usually, really concerned about that the risk and benefit profile, and also the study is designed in a way to allow a valid statistical analysis so you can make conclusion whether something is safe or effective or not. Of note is that most for a new chemical candidate, usually only about 10% success rate after entering into clinical trial testing. And the clinical trial or the purpose and procedure, and we'll come to that, is clearly defined in a clinical trial protocol. Right? Now, a few terminology on the clinical trials. One, randomized. That usually means the study participant is randomly assigned one of the two study groups that are in the clinical trials. There's open label or blinded study. Open label, these two are opposite to the blinded. Blinded usually means someone is masked to the treatment assignment, and that could be the investigator that's masked or the study participant that's masked. And when the double blind usually means both the investigator and the patients are both masked to the treatment assignment until the study is over. You might have hear about a single arm study or open label study, and the single arm study, mostly open label study. It does come with the inherent risk because you have not another group to compare whether something is safe or effective or not. And there are a concept of control versus uncontrolled, and that relates to the comparator groups. So today, the randomized double-blind placebo or the control trial, or called RCT, is really the gold standard clinical intervention drug, in this case that we're talking about here, for both safety and effectiveness. Another things that you often hear about are different phases of clinical trial, from phase 1 to phase 4. And this is in part because of the stepwise process to evaluate the safety profile, and that was increasing the longer duration and more study participants. So phase 1, safety and safety first in human, and is usually also includes a pharmacokinetic assessment going from single to repeat dose study. Sample size usually small, and is usually occurring, the study is conducted in healthy individuals. Occasionally it can be in patient population as well. Moving on to phase 2, again, more on safety, but also phase 2 studies seek to find evidence of initial evidence for that whether the drug can work or not, and also at what dose can it work. Study is usually longer than phase 1. There are more people, and phase 2 is usually conducted in the intended patient population. Phase 3 then is something we call confirmatory trial on safety and effectiveness, and usually is a control trial or with a comparator arm, whether that comparator arm is a placebo, the current standard care, or an active intervention. More people for sure, and usually, and again, is studied in the intended treatment population, patient population, for a longer period of time. And how long it is, it depends on the intended treatment duration for that disease, for that therapy. Occasionally, you may hear about the phase 4, and that usually is post-marketing trials. These are the general framework, phase 1, 2, 3, but sometimes there are variations, can be combined, or can be split further into 2A, 2B, or even combined phase 1, 2 study. Let me just go back on the safety and how that's done, and using phase 1 as the illustration. For example, you might have a new therapy that you want to evaluate. You think you can go for about 30 to 100 milligrams. That's likely the quote-unquote clinical dose. But when you first started in the phase 1 study, you will not go straight to that 30 to 100 milligram that you think is going to be producing the effect, you actually would be very carefully starting with only a fraction of that 30 milligram and go stepwise, in this case, a threefold increase, starting with the 1 milligram, moving up, evaluating each cohort and understand the safety profile before you making the move into the next cohort of a higher dose. And that's why we call a single ascending dose study. Right? Now, besides these clinical trials, there are also another set of clinical study that's done, and we call it the clinical pharmacology or biopharmaceutical study. And these are mostly focusing on assessing the pharmacokinetics and the ADME profile of the drug, and usually it's done in healthy individuals. And that can include also the pharmacodynamics and the effect of different people might have with these PK, and that includes whether the genetic background, the sex, the age, or the ethnicity, or their different organ dysfunction, someone with kidney dysfunction or someone with the liver dysfunction. It can also evaluate the drugs or food that we take. Does that have an effect on the absorption of these medications? And you know, some of the examples of the different studies that are listed here. Now, let's say when we are ready to go into clinical trials, we obviously need a clinical trial protocol. And this is a document that provides the background and rationale of conducting a trial. There are many components in here, but particularly and specifically why we're doing it, what are the scientific rationale, what are the risk-benefit assessment, what's kind of design, and why that design specifically, the intervention that we're going to provide or evaluate, and the duration, the study population, that is the inclusion-exclusion criteria, and what kind of assessment will be done, and also the statistical analysis considerations. Right. So, and this is an example of the protocol content, and you can see there are a lot of information in there, and the study trial protocol can easily run to 100 pages or more. So who contributed to these writing or preparation of the clinical trial protocol? And these can come from the sponsor or the investigator if the investigator-initiated study, but usually involves a large multidisciplinary team, going from representative from clinical function to clinical pharmacology, safety or pharmacovigilance, biostatistics, clinical operation, and so on and so forth. There's obviously, in most situations, beyond these people preparing for it, there's a specific protocol review committee that goes through and approve that protocol. The principal investigator of the study simply contribute to it, but the FDA or the ethics committee also contribute to commenting on review of the clinical trial protocol before the study can be initiated. And once you have that protocol, can you start? No. Particularly for investigational drugs, you need to file what's called an IND or investigational new drug. That could be a new chemical entity or for a new indication in a different target population. The IND approval is necessary to start the trial, and the IND could be investigator IND or the company-initiated so-called commercial INDs. These are highly regulated, and it's under the Code of Federal Regulation. When you apply for IND, besides the protocol, there are a lot more information that need to be provided, and some of them are listed here. In Europe, the process is not IND, but what they call CTA, the clinical trial application. And currently that's under the regulation of clinical trial registration process. in the European under the European Medicine Agency's authority. Now, a few more words about these regulatory authority. In U.S., FDA is the major government agency. The FDA was first created to enforce the so-called Pure Drug and Food and Drug Act of 1906. In Europe, EMA, as I mentioned, is a counterpart, and in Japan, PMDA is the regulatory agency that review and the clinical trial and the new medicine development. For those of you who are the history buff, just a note of the creation of FDA has been accredited to Dr. Harvey Wiley, who started as a chemist and is referred to as crusading chemist because of his relentless effort in promoting the food safety and later the drug safety. Now, beyond the regulatory agency I mentioned, there's the IRB, or the Institutional Review Board, and in Europe it's called the Ethics Committee or Independent Ethics Committee. This is a group of experts, a multidisciplinary team who formally review, approve, and monitor human research subject to make sure that their right, their safety, and their welfare are protected. IRB are often tasked to determine the degree of risk and whether the risk is acceptable or not. In a clinical trial, they may have a local IRB, may have a central IRB, or may have both a local and central IRB, or more than one local IRB, depends on how many study sites, how many different countries that particular clinical trial engaged in. There's an effort on something called small IRB to harmonize these processes to make and streamline so that it doesn't become too overtly burdensome. So beyond that, beyond the approval from the FDA or Ethics Committee, there's a further disclosure necessary to the general public, because the public, and particularly the patient community, is clearly a key stakeholder in the clinical trial process. In the US, there is a clinicaltrial.gov that's under the auspices of the NIH or the National Library of Medicine, and the study has to be registered, information updated, and this regulation is really not just by federal law, but also by the journal publication standard and potentially by funders as well. In EU, it's under the EU clinical trial registered process, right? So once you have all that, you can start the clinical trial. And who are conducting the clinical trial? And that's the people called investigator or principal investigator, who is responsible for the overall conduct of the study at the site, with the specific remit to protect the right and welfare of the humans, study participants, and also to ensure the validity and integrity of the data collection following the study protocol. They have a lot of roles and responsibility. A few of them listed here, and this is only the highlight. Besides taking care of the patients in the study trials, in the conduct of the study, they supervise the staff. There are a lot of work that they need to do and communicate with the regulatory bodies such as the IRB. And with all this, obviously, an investigator cannot do everything, and neither a staff supporting that, and the investigator can delegate to. So at the site, you also hear about the research nurse, the study coordinator, the pharmacist, or even the lab technician or the phlebotomist, and there are other regulatory coordinator. Because PXE is a multi-organ systemic disease, if you really want to evaluate the outcomes endpoint that may involve more than one organ system. And most of the physicians are specialized. So you might have a principal investigator. You might also have, let's say that investigator is a dermatologist. You might also have investigator that's also ophthalmologist or a vascular person or other type of clinicians. There's also, it could have additional sub-investigator in that side as well. Now, so beyond the investigator, who else is there? Obviously, there's you, the trial participants. What can you expect out of the clinical trials if you were to participate? Certainly, you want to be informed of the clinical trial. Why is it being done? What is the trial? And what kind of treatment is being evaluated? And how much involvement do you need? Right? Whether that's the study procedure, the time commitment, the travel requirement, or that the confidentiality is given to you, maintaining your confidential information, and what the role of social media, all those things, you really want to be informed. And this document is called informed consent that you need to sign, but it's not the form itself. It's really the critical process that make you understand why you may be interested in participating in clinical trials. So that process itself is what's important for the informed consent. Simply at the end, you need to sign it, but also important to know that you can withdraw your consent at any time during the study. In the study itself, you'll go through a screening period that consists of interview, exam, and test procedures. And during the study, there will be a so-called on-treatment assessment while you're taking the prescribed study medication. And those assessments include a multitude of things: exam, blood tests, ECG, or other imaging procedure, or clinical procedures to evaluate how well you do with whichever arms of the study you're in, placebo or active treatment. There will also be the post-treatment assessment as well. And I want to note that the current clinical advances that we have today really are all thanks to the participant in the clinical trials, people like you. Right? So I mentioned a few times, ensuring clinical safety is very important, and it is in clinical trial and in medicine development. We usually characterize this by something called adverse event, and that means anything untoward that happened is captured, and the capture is adverse event. We think of those events and then characterize them as in different severity, or is it a very serious or not serious event? There will be causality assessment, and particularly in study with control group. That's important for comparison purposes. Adverse event can be common or could be rare. And that's why we need to think about the size and duration, moving from phase 1 being small and shorter to phase 3 being larger and longer duration, to be able to capture the event that are increasingly having the ability to capture events that are more rare. Right? And who during the clinical trial process monitor for these safety? I mentioned about the regulatory agency, the ethics committee or the IRB. There's obviously the investigator at the site will watch out for it, from the sponsor and the research contract research organization will come back to in a second. There are medical monitors that do that from both organizations. And there are also something called steering committee and data safety monitoring board. Let me touch on those two entities. The trial steering committee, or steering committee in short, really provides a trial oversight. And these are typically organized by the sponsor, and it's independent to a large degree of the oversight committee. They are charged to monitor and supervise the progress of the study toward its objective. They review the progress on the regular DSMB about the assessment of risk. Okay. The DSMB then, what's called Data Safety Monitoring Committee, and this is a group that organized specifically to monitor for the continued trial participant safety while maintaining the trial integrity. You may hear a different acronym, DSMC, DMC, or DMB. And these consist not just only of clinician, but also statistician, occasionally, depending on the trial need, may involve also bioethicists or patient advocates as well. And the DSMB really need to be operating independently and separate from the trial investigators, and maintain a high degree of confidentiality. So with all those I just described, all the activity that need to occur, it's a lot of work, right? And the sponsor, either the investigator or biopharmaceutical company sponsoring the study, cannot do everything. And they often hire a CRO, or so-called contract research organization, to help execute the study. And that includes activities such as project management, the submissions, the site management, all the way to the back end, trial monitoring, the medical writing, and so on and so forth. In the trial, there are also other groups, such as laboratory that I used, and that's a different group of people that need to run the test, handling the management, analysis of biological sample, imaging information. You may hear a word of central lab that usually perform the lab tests that are routinely used in clinical practice. There may be a specialty lab that handle mostly tests that are not yet routinely used. And there could be a new assay, new biomarkers, and whatnot. The core labs that usually specialize in uniform analysis of endpoints that are different than the simple blood test. And that could be imaging results, could be cytology, could be EKG, could be pathology samples. Separately, there's genomics lab if we're doing genotyping analysis, right? So and lastly, there are other trial support groups that may come into play in a clinical trial. That includes home visiting nurse, particularly in the age of virtual clinical trials. There may be travel services, maybe pharmacy service, or maybe a particular group that handles so-called smart devices for the virtual study procedures or visits. So this takes really a lot of people to take care of this and to implement and doing the study operation activity. In the CRO team, it would not be surprising to have 20, 30 or more people on the study team to handle the various activity that going from the medical monitors to data coordinators to home health services or clinical safety. And similarly, these roles and responsibility also occur in the sponsor's team, whether that's the pharmaceutical company or other biotech companies, right? So this coming to the end here is for clinical trial, right? We have participant, you, the investigator or the site staff running the clinical trials with the help of PX International for these PXE trials. There's a sponsor that are organizing this, but there are many more people involved in the process of running clinical trials. As I mentioned, there's certainly the sponsor and CRO study teams supporting the investigator and supporting the trial and supporting the trial participants. There are also the IRB that are intimately involved in every step of the way. And to getting the study from the beginning, conceptualization to running the trial, There's the other expert consultants, advisory board, steering committee, DSMB, as I mentioned, and occasionally there may be event adjudication committee to make sure that data collection and interpretation of the endpoints are uniform. There are the laboratories, many different labs. There are also the pharmacy, the agent, travel agency, home health nurse, and there's IRB and the regulatory authorities, and there and also the posting, the clinicaltrial.gov. And all this really coming to the end is that it really takes a village. It takes a PXE village to run these clinical trials. No one can do it alone. And quoting from Helen Keller, an advocate, alone, we can do so little. Together, we can do so much. So thank you very much, particularly the people with PXE and their family, the PXE scientific and clinical research community, Sharon, and also the PXE International's folks. Thank you very much. PXE medicine development, it certainly takes the PXE village. Thank you. Now I'll take a few questions.
Thank you very much, Hubert, for your presentation. I'll add a little that sometimes there have been clinical studies that have not had a pharmaceutical company or even an academic investigator involved directly or primarily. PXE International has run a number of those studies. Some of you have participated in the very early studies where we drew your blood to even see, did your pyrophosphate levels vary in your blood? And then certainly the long-term epidemiological study that you've all been involved in as well. And all of the things that Hubert said apply to those as well. Okay, so questions. I'm going to take your slides down, Hubert, so people can see you. Questions. You can either use the raise your hand function on Zoom. If you can find it, that's a little tricky. You can unmute. You can put a question in the chat, your choice.
Hubert, what would be some reasons that we wouldn't get to a phase 3 trial?
So I think you're referring to once a particular project enters into phase 1, there may be only a 10% chance finishing phase 3 or registration. There are many reasons, and really it's coming back to safety and effectiveness. Many of drugs may have safety signals that were not identified, or from the preclinical study. And that can be one big reason, not able to find the right dose or the effectiveness that we're looking for, either whatever the endpoint of the phase 3 trial may not be, say, is not suitable to develop the not effective enough by whatever measure of using the endpoint for that trial. So those are typically the two large areas of reason. Sometimes occasionally a smaller percentage, it could be the opportunity for, let's say, in the setting of a biopharmaceutical company, they decided to invest somewhere else. That can happen as well. But usually there may be, in those settings, they may out-license to a different company that may be interested in.
And in common conditions, the number one reason is low enrollment, that they can't get enough people to participate in a trial, even after trying for many years. Yeah. But not so in PXE.
That can happen, particularly, a challenging situation for rare disease indication. It may be more, it is definitely much more challenging.
But we've been very pleased with all of your responsiveness. And I didn't say at the beginning, but please do not identify whether you've been in any PXE trials, because the sponsors can't know that. Any other questions? We'll take one more, because then we have to close this link and go to the next one.
I have a question.
Yes.
Thank you so much for your presentation, truly. My question is, what trials, I mean, can you let us know what trials are happening now, or did you and I missed it? And the other thing is, yeah, what trials are happening now, and how can we help? Thank you.
I'm pretty sure, in large part, I would say PX International has been very good in terms of the PXE trials and something
Yes.
of the genetic alliance part for sure. But the clinicaltrial.gov website that I mentioned is one good place, and that is exactly intended for potential patients, family interested in knowing what clinical trials are ongoing for any particular disease. So that's a very good resource for it. If you want to find out what's being and I would say both the academic investigator and also from biopharmaceutical company launching a clinical trial, they are required to disclose that on the clinicaltrial.gov in EU, the clinical trial registry. So that's a good place to find information.
Okay, we're gonna wrap it up. I put clinicaltrials.gov in the chat, so you can click it there, or we can send it to you if you can't get it out of there. And as Hubert said, we announce the trials even in advance of when they are formally registered. If we can, you always know what's coming down the pike through the PXE International newsletter, which you get every single month. And if you don't get it, then email mary@pxe.org. And she will see what's wrong with your email address. Hubert, I want to thank you very much for presenting today. This was a great overview. It goes into our archive, and so we'll be able to point to it when people ask us in all of our various places like Facebook and on our Google group, what is a clinical trial? This is really a fabulous presentation on what people can expect and know about clinical trials as we move through this very important phase for PXE. So thank you. And we really appreciate working with you.
Thank you, everyone.
Thank you.